Okay, with the establishment of genetic engineering technique, where it was possible to isolate, okay, and cloning or clone, okay, any piece of DNA, and availability in simple and fast techniques for determining DNA sequences. Okay, very ambitious project of sequencing the human genome was launched in the year 1990. Human Genome Project was called as a mega project due to its extraordinary hard work, or its a structural framework. Human genome is said to be have approximately 3 into 10 to the power of 9 base pairs. Here it is mentioned as 10 to the power of 9 base pairs. Estimated cost of sequencing required US dollars, $3 per one base pair. Total estimated project would be approximately $9 billion. Okay, US dollars. Further, if the obtained sequence were to be stored or typed, okay, after typing in the computer and making them to store or converting into books by printing methods, if each page of the book contained 1000 letters, each book contained 1000 pages, then 3300 such books would be required to store the information of DNA sequence from a single human cell. Okay. Now here the enormous amount of data expected to be generated at a high speed computational devices. Separate languages are required. Separate data storages are required to store this information. HGP was also closely associated with the rapid development of a new area in biology called bioinformatics. Human Genome Project, some of the important goals are mentioned here as follows. Identify all the approximately 20,000 to 25,000 genes in human DNA. Determine the sequence of 3 billion chemical base pairs that make up human DNA. Store this information in database. Okay. At the same time, import tools for data analysis. Transfer related techniques. If you wanted to transfer this data from one industry to or another industry, or from one device to another device, and at the same time it addresses ethical, legal and social issues that may arise from the project. The Human Genome Project was 13-year-old project coordinated by the US Department of Energy, National Institute of Health. During early years of the Human Genome Project, a Wellcome Trust of UK became a major partner. Additional contributions came from Japan as well as France, Germany, China and others. This project was completed in 2003. Knowledge about the DNA variations among the individuals, okay, it becomes revolutionary, new ways to diagnose, treat some genetic disorders, to prevent the thousands of genetic disorders. So DNA sequencing lead to an understanding about the genetic information, natural capabilities. Okay. Next, many non-human model organisms such as bacteria, yeast and Caenorhabditis elegans, Drosophila, plants, etc. also be sequenced. The methods involved a major approach. One approach focused on the identifying all the genes that are expressed as RNA, referred to as express sequence tags. The other took the blind approach of simple sequencing the whole set of genome that contained all the coding, non-coding sequences. Okay. Now in this case, sequencing the total DNA from a cell is isolated and converted into random fragments relatively smaller in size. Recall, DNA is a very long polymer and is cloned in suitable hosts using vectors. You know very well about cloning. In biotechnology, I will explain what is cloning. Cloning resulted into amplification of each piece of DNA fragment so that it is subsequently could be sequenced with ease. Commonly used hosts were bacteria. And yeast. The vectors called as bacterial artificial chromosome and yeast artificial chromosomes were also used in this sequence. You can see how a human genome was collected from a specific cell, extracted the genetic material and converted into computer-based data with the specific languages. Fragments were sequenced using automated DNA sequences. They worked on the principle of developed by Frederick Sanger. Okay. Remember, Sanger is also credited for developing method. These sequences were then arranged based on some sequences overlapping regions present in them. This required